Okay, so good afternoon, everybody, and welcome to the last plenary talk for today's session. It is a great pleasure to introduce Dr. Barry Scott. Dr. Barry Scott is a professor of molecular genetics at the School of Fundamental Sciences at Massey University in Auckland, in the North Island of New Zealand, where he was appointed in 1985. He obtained both his Bachelor of Science and the Doctor of Philosophy degrees at the University of Otago in Dunedin, which is in the South Island of New Zealand. He is an emeritus investigator in the Bioprotection Research Center, a national center of research excellence, and he held the position of Vice President of the Royal Society, I don't know if I'm going to say that right, Te Aperangi Council from 2015 to 2018. He was also head of the Institute of Molecular Biosciences for 5 years. And he was a founding board member of the New Zealand Genomics company, which he currently directs, and also of the Environmental Risk Management Authority. He has published over 120 articles and directed about 21 PhD theses, and has received many awards. Among them, I think two of the most prominent is that he was elected Fellow of the Royal Society of New Zealand in 2010. And in 2014, he was awarded a Humboldt Research Award from the Alexander von Humboldt Foundation of Germany. His major area of research is fungal plant symbionts, and we'll hear about that in his talk, I hope. But early in his career, he made landmark contributions to the understanding of rhizobium legume symbiosis. His group is interested in understanding the molecular and cellular basis for establishment and maintenance of the symbiosis between the grass endophyte Epichloe festucae and perennial ryegrass, which is economically important to New Zealand agriculture. He has identified the endophyte genes and pathways that are responsible for the biosynthesis of secondary metabolites that are unique to the symbiosis. And another of his major contributions was the demonstration that fungal synthesis of reactive oxygen species is essential for the stable maintenance of the symbiosis. So without further ado, I would like to give the microphone to Barry. Thanks again for accepting our invitation. I'm looking forward to hearing your story. Thanks, Barry.
Thank you, Meritzel, for that introduction, and thank you, Edgardo, for your correspondence. And welcome, everyone. I think this is probably the first talk I've ever given distantly, so it is a bit strange for me talking from my office here in Palmerston, North New Zealand, to you people in Mexico. So I hope you've had a great symposium. It's the end of the day, so I hope I can keep your attention. So for quite a long time, I've worked with the symbiotic interaction between a filamentous fungus called Epichloe festucae and a grass called Lolium perenne. This is ryegrass. And I think it's probably fair to say now that this is the most well-developed fungal plant aerial symbiosis. So we know a lot about rhizobium and interaction with legumes. We know a huge amount about Arbuscula mycorrhiza and roots of plants. But in terms of aerial symbioses, it's a relatively unexplored area. And one thing I've really enjoyed is developing the tools to understand interaction between this leaf symbiont and this grass. So Epichloe festucae, I won't go through this life cycle in detail, but just to say that it has an asexual life cycle. It lives in the intercellular spaces of the leaves of the grass. It doesn't break through the cell wall into the plant. It just grows intercellularly. So this is part of the mutualistic interaction. And when the grass flowers, it colonizes the reproductive tissue into the seed falls on the ground, the seed germinates, and it's carried through from one generation to the next. There are a lot of asexual species that have no other life cycle, so they're permanently in grasses, and these tend to be mutualistic and beneficial for the association. There are some species, including Epichloe festucae, on certain hosts can, when the plant undergoes reproduction, switch from a very restricted growth to proliferative growth, and they break through the leaf surface to form the stroma or sheath around the flag leaf of the grass and essentially prevent the flowering meristem from emerging, so they choke the grass. And then there's a little fly that can transfer a spore from one stroma to another stroma. This is an outcrossing species, to initiate meiosis and the sexual life cycle. And from that, you get ascospores, which can reinfect flowers, recolonize the leaves of the grass to complete the sexual cycle. And this is an image, courtesy of Jim White from Rutgers. It's a lovely image of Epichloe festucae growing on fine fescue. And what you'll note here is the dramatic transition from the substromatal tissue to that stroma. And this coincides precisely with where the flowering tiller is located. So we assume that when the plant transitions into the reproductive cycle, all those nutrients flowing to the flowers trigger formation of the stroma. And the ascospores on this white surface are carried by the insect from one stroma to another to initiate the sexual life cycle. And you get a change in the color of that stroma from white to orange, coinciding with the formation of carotenoids. The interaction we study and we've developed over many years is between ryegrass and Epichloe festucae. And so there's a ryegrass plant on the left, and if you take a leaf and stain it with aniline blue, you see these little railway tracks of hyphae growing through the intercellular spaces of the leaf and horizontal to the direction of growth. If you take a transverse section and look under the electron microscope, you'll see those single hyphae are physically attached to the cell walls of the plant. And this becomes important, and I'll come back to this in a moment. If you introduce GFP into the Epichloe and then infect a grass seedling and grow it up, you'll see this amazing network of hyphae growing through the cell division zone and up into the leaves and developing leaves of the aerial tissue of the grass. Importantly, this particular fungus doesn't grow by tip growth in the leaves, it grows by intercalary extension. This was work done by the AgResearch group here in Palmerston North many years ago. And this is quite remarkable because this is exactly how leaves grow. When you mow your lawn, you cut off the leaves off the top, but the meristem is at the base. That's where the cell division occurs. And then you get the cell expansion. And if you have hyphae that are physically attached to those cells, as those plant cells expand, they stretch the hyphae, and that stretching triggers intercalary division and growth. And so the growth of the fungus is very nicely synchronized with the growth of the plant. So in summertime, when your lawns are growing very fast, the hyphae are growing equally fast, about a centimetre or two a day. And so this is a very different form of growth and very, very interesting. If you take some leaf tissue and stain it, infiltrate it with aniline blue, which stains beta-glucans, and Elixiflor 488, which will stain chitin, what you notice is you see these long stretches of hyphae, but just the septa staining up with the Elixiflor wheat germagluten 488. Now this is a lectin that binds chitin. And so when we first developed this technique, I was surprised that the cell walls of the hyphae were not lighting up with this chitin-specific stain, but just the septa. So we wondered for a long time whether there was some change in the cell wall structure during endophytic growth. And I'll come back to this in a moment. Not only is Epichloe festucae an endophyte, it's also an epiphyte. It can form an aprosorium inside the plant and using turgor pressure, break through the surface and then form a network of hyphae on the surface of the leaf. And this is a lovely image captured by Becker back in 2016. And this is showing the leaf surface just above the of the plant. One of the things we've done is to do both transcriptomics of the plant and the symbiont, the fungus, and using computer methods to deconvolute one transcriptome from the other. In 2015, we looked at how the plant transcriptome was affected by the presence of the fungus. So comparing plants with and without endophyte. And not surprisingly, you see this dramatic change in the transcriptome when you infect plants with Epichloe festucae. And I won't go through all the detail. This was published in New Phytologist back in 2015. But the fungus totally reprograms Lolium perenni metabolism. It also affects plant development, and just two examples in this paper are the cell walls of infected hyphae. This is the plant cell walls, are thinner than plant cell walls without the fungus present. And another interesting observation is the ligules tend to grow, the trichomes on the surface of the leaf grow slightly bigger in infected plants versus uninfected plants. So these are very interesting observations. The biochemical explanation of these remains to be studied. And of course, that's something for the future for you younger scientists. Oops. Can you hear me?
Yes, no, yes. Mm-hmm.
Sorry, my slide seems to have frozen. I'll just not quite.
You can maybe, yeah, there you go.
Yeah, it's fine now. So when Epichloe festucae is grown in culture, if you look at the transcriptome in culture versus the transcriptome in planter, not surprisingly, there's a lot of gene expression changes. So this is also a comparison we've made. And you see of the order of about 1,000 genes that are differentially expressed in the fungus between free-living culture and in planter. And many of these are, not surprisingly, genes involved in the expression and synthesis of secondary metabolite pathways, a lot of effectors, gene products that affect or suppress host defense, these sort of interactions in the plant. But it's the secondary metabolite genes that are among some of the most dramatically expressed. And just two examples here, genes involved in the synthesis of lolitrims, these are indole diterpenes, and genes involved in ergot alkaloids. These are clusters of genes that sit on the end of chromosomes 1 and chromosome 3 of the Epichloe festucae genome. We have a whole complete genome sequence now, and we know exactly where all the genes are. But these are interesting because if you look at the expression of these genes in culture, they're totally shut off. And you can see in image B, I won't go through the detail too much, but if you use GFP hooked up to one of these promoters, you can see that the promoter is not expressed in culture compared to a constitutive promoter like GPD, but in plantar, that promoter is very active and you get expression of GFP. And so these are genes that are specifically expressed once the fungus colonizes the plant. Again, we're not entirely sure what triggers this. There's some major epigenetic changes. We've published on that, but exactly what the trigger is for this transition still remains to be studied. And this is just an image showing that Epichloe has 7 chromosomes. Those 2 gene clusters I mentioned, the ergot alkaloid cluster that's on the end of chromosome 1, the indole-diterpene or lolitrim clusters on the end of chromosome 3, and the other little boxes there just show many of the genes that we've studied over a long period of time and where they're located. And I must say it was quite exciting to have a fully assembled genome and to be able to go in there and see where your favorite gene was located and see the context in which that gene was located, whether it was repeat sequence and things like that. Now, these bioprotective metabolites, I don't have time today to talk too much about them, but these are important part of the symbiosis. So the fungus produces many different metabolites and it's very strain species dependent. And we keep discovering more each year. But some of the ones that are best known and best described include on the left a non-ribosomal peptide synthetase product called pyramine. This is potent anti-insect activity. Lolines, which Chris Shardle at University of Kentucky studied, these are potent insecticides. And then on the right, you have lolitrim B, which is an indole-diterpene, and ergovaline, which is an ergot alkaloid. And these 2 classes of compound are mammalian toxins. So in natural plant ecosystems, these are beneficial for the association. But of course, when you move these grasses from natural ecosystems into agricultural ecosystems, then you have problems for stock that are grazing on these grasses where these compounds are made. And so there's a lot of agricultural interest in these endophytes and finding strains that make compounds that are beneficial for the protection of the plants from insects, for example, but not toxic to animals. And there's a whole lot of work and commercial development around that. And this is just showing you an image of Argentine stem weevil on the surface of the leaf and the structure there of pyramine. And what's interesting is that Argentine stem weevil, when it sniffs that compound, it deters it from feeding on the grass. And again, there's some fascinating biology still there to be unraveled as to how that insect detects that compound, what are the receptors for it, and so on. But what I want to talk about today is some recent work, and this comes back to the cell wall structure of this endophyte when it's growing inside the grass. I'm just showing this image again of the leaf stained with aniline blue and alexiflor wheat germ agglutinin 488. And as I mentioned before, the wheat germ agglutinin only lights up the septa, as you can see there in the turquoise color, but not the cell wall. And so I wondered whether what was going on was that chitin, which you find normally in the cell wall of fungal cells, is being converted to chitosan. There are other hypotheses, but this was the hypothesis we wanted to test. And how we went about testing that and was to try various chitin and chitosan biosensors. I've mentioned already the wheat germ agglutinin AF488, which is well known, binds to chitin. But in collaboration with Bruno Muschbacher in Munster, we got access to 2 other biosensors, one a chitin binding protein hooked up to GFP, and you can see there the details, it's from a bacillus, and a chitosan affinity protein also hooked up to GFP, again from a bacillus gene. We also got from another group in Denmark, OGA 488. This is an oligogalacturonate that binds chitosan. So then PhD student Nazanin Nourifar set about for her PhD to test these different biosensors on Epichloe festucae when it's grown on the surface of the leaf, inside the plant as an endophyte, and on the surface as an epiphyte. And what she found, and what we have to do, this fungus doesn't naturally just infect plants, is you have to make a little cut with a scalpel, and this is showing the insertion site. And you can see there on the left the brightly blue fluorescence of the hyphae growing outside on the surface of the plant. And there in figure B in the center, you'll see little with white arrows pointing out where the hyphae are. You can see the septa lighting up, but the cell walls are no longer fluorescing with the wheat germ agglutinin. And this is highlighted in C and D. You can see in C, the hyphae on the surface fluorescing brightly, whereas the hyphae inside the endophytic hyphae, just the septa are fluorescing. So this suggests that there's a dramatic change in the cell wall from having chitin present outside to loss of chitin once the endophyte colonizes and is endophytic with inside the plant. And using the chitin binding protein, we confirmed that endophytic hyphae have chitin, at least that's accessible, just in the septa and not in the cell wall of endophytic hyphae. And this is work that's online now in molecular plant microbe interactions. Whereas if you look at hyphae in the middle image there that have emerged from the endophytic state to start growing on the surface, you see a little region there labeled X for expressorium. This is where this aprosorium-like structure forms. As the hyphae come out of the leaf, you see this transition from chitin just in the septa to chitin being restored in the cell wall. So when the fungus breaks out and starts growing on the surface, you get this transition back to chitin present in the cell wall. So loss of chitin when you infect to from endophytic hyphae, restoration when it grows on the surface. Nazanin also investigated using the chitosan affinity protein, and this was quite tricky to work with because it's a big protein and it took quite a lot of work and playing around to get it nicely infiltrated into the tissue. But if you look across the images A, B, C, and D, you can see that this particular biosensor is labeling up the cell wall. And using propydium iodide, you can see the nuclei there fluorescing in red, but no sign of septa. So this chitosan affinity protein is binding the cell wall, suggesting there is chitosan in the cell wall, but it's not binding to the septa. So this is the reciprocal of what we see with the wheat germ agglutinin 488, strongly suggesting that our hypothesis is supported that endophytic hyphae have chitosan in the cell wall and chitin in the septa. The OGA 488 was more difficult to work with, and it seemed to be much more diffuse. But what you will note, where these white arrows are in F, that there's no binding to the septa, again supporting chitosan in the cell wall absent from the septa. And if you look in the cell division zone again with this chitosan affinity protein, you find that again, you find the hyphae cell wall lighting up with the chitosan affinity protein, propydium iodide showing the nuclei there of both the plant and the smaller nuclei of the fungus, but again, absence of septa. So just these long strands of the cell walls of the fungus lighting up with the chitosan, providing further support for chitosan formation. So many of you will know that chitosan is formed from chitin when you remove the acetyl groups, as shown here, from the N-acetyl glucosamine residues that make up chitin in the cell wall. So here's a typical cell wall structure of a fungus: cell membrane, chitin, beta-glucans, monoproteins, and this chitin has a structure of N-acetyl glucosamines. If you lose those acetyl groups, you form chitosan. And the enzyme that does that is a chitin deacetylase. There's been a lot of papers published on these enzymes recently, and many fungi have quite a large number of these chitin deacetylases. Epichloe festucae has just 3, which we have named CDA-A, CDA-B, and CDA-C. And if we look at our transcriptome data, you will see that the CDA-A is differentially expressed, 7-fold difference when the fungus is grown in planter versus in culture. CDA-B, not really differentially expressed at all, even though it is quite well expressed both in plant and culture. And CDA-C essentially not expressed at all. So based on this analysis, we thought we'd delete the gene for the CDA-A because it was differentially expressed. And what we always do is have a look first whether the phenotype of the fungus changes in culture. So this image here shows you the wild type FL1 and 3 independent mutants of the CDA gene. And to cut a long story short, the hyphae, this is staining with calcifluor white, the hyphae grow the same, have the same morphology and growth as the wild type. They form these spectacular coils, they undergo cell-cell fusion, but deletion of that chitin deacetylase does not appear to have any impact on the growth of Epichloe festucae on a Petri dish in the lab. If you take that mutant and inoculate into ryegrass seedlings, and this is just an image showing you how it's done, you make a little slit in the hypocotyl of the leaf, insert a little bit of mycelia, and then allow the seedling to grow, and that mycelia will colonize and infect the entire plant systemically. And then after about 12 weeks, you can the seeds, and shown here are some seedlings infected with the wild type and 2 independent mutants of the CDA-A gene. And what we normally do is measure the tiller number and the length of the leaves to see if there are any differences. And as you can see on the right, there's no significant difference, although there did appear to be some smaller plants in some of these experiments, like shown here with CDA-A, but not statistically significant. However, if you took the leaves from those mutant plants and then stained them up with aniline blue and wheat germ agglutinin, you can see here there's a dramatic change in that intercalary growth of the hyphae within the plant. So there's 3 panels here on the left for FL1, the wild type, A, B, and C, just showing that the wild type grows as nice parallel strands of hyphae through the grass leaf tissue. And the 3 independent mutants, well, you can see the hyphae are all over the places. Most dramatically, the septa are very, very close together, suggesting there's a lot of cell division, but that intercalary growth is disrupted. So although you don't see a whole plant phenotype, there definitely is a cellular phenotype with this mutant. So we thought we'd better look at the CDA-B mutant as well and see what impact that has, as well as the CDA-A and CDA-B double mutant. And the CDA-B mutant, even though it's not differentially expressed, had quite a dramatic phenotype. And you can see here the seedlings in the top image are quite a lot smaller than the plants infected with the wild type. And the height of those leaves was statistically significantly different between the mutant and the wild type. But interestingly, and a little bit puzzling, one might say, in the double mutant, the difference was not so dramatic and was not statistically significant. And we had a little bit of difficulty here with the referees, because one referee said, well, you know, this is strange. How do you explain this? And I had to write back and I said, well, you know, as we know frequently, sometimes science is just a little bit complex. It's not what you expect. And so you have to explain it. And so one explanation, and I have others in a moment I'll come to, is genetically the CDAA mutant must be dominant to CDAB and to have this particular phenotype. I'll come back to some possible explanations in a moment. But as with the CDAA mutant, when you look at the cellular phenotype, i.e. how the fungus is growing inside the plant, you can see again that the intercalary growth with the CDAB mutant is also disrupted, as is the CDAAB double mutant. If you look at this image right in the middle here in row B for CDAB, I mean, it's quite remarkable, all these stacks of hyphae on top of one another. It really is growing in quite a bizarre way inside the plant. And importantly, when you do these genetic analyses, you need to demonstrate that the mutation you've made is responsible for that phenotype. So typically what we do is reintroduce the wild type gene, and we did this for both the CDAA mutant in column 3 and the CDAB mutant in column 5 here. And you see restoration of the cellular phenotype when you put that wild type gene back in. So this confirms that those mutations were responsible for those phenotypes. So why does the CDAB have a more dramatic whole plant phenotype than the A alone or the AB double mutant? So Jonathan Luquito, another student in the lab, did some qPCR, relative analysis of biomass, and found that the CDAB mutant had more endophyte biomass, both in the pseudostem, that's the lower part of the leaf, and the blade, whereas the CDAA mutant and the double mutant had very similar biomass to the wild type. And we found this with many mutants, that where you get more growth, more biomass, there's an impact on the growth of the plant, and you often see this plant stunting. What's a little bit unusual with CDAA and the double mutant is that there is a cellular phenotype, but the biomass overall, when you do qPCR, is very similar to wild type. So you've got to be careful in looking at those images and thinking there's a lot more hyphae there, and you need to actually measure it. And this graph, this analysis here shows that the biomass is in fact for A and the double mutant the same. Oops. So what about, or the other thing we needed to explain was, if we go back to, it's not behaving for me. If you go back to the slide here and look at the CDAA and the double mutant, what you notice is that with the wheat germ agglutinin 488, you still don't see restoration of chitin in the cell wall. There's chitin in the septa. So the two chitin deacetylases, even when you knock both of them out, you still don't seem to see restoration of chitin in the cell wall. So this is one of the more puzzling results that's been a little bit difficult to explain. And of course, the referees asked, well, what about CDAC? So we did RT-qPCR on all 3 genes. And what you find is this gene is essentially not expressed. And so the presence of that 3rd potential gene is not the reason. And so we're still trying to figure out exactly why restoration of chitin does not occur in the endophytic hyphae when you delete both of these genes. And further work is required on that. Of course, when you make these mutants and the growth is disrupted, you may be affecting a whole lot of other things, and chitin is just not restored. And just to finish off with an interesting thought, is not long after we submitted this work, I got an email from Jean-Michel Ahn in University of Wisconsin. And they have just published this paper earlier this year in Nature Communications, showing that many fungi make oligosaccharides, similar to the lipo-oligosaccharides made by Rhizobium, which many of you will know, a nodulation signal sent by Rhizobium to the legume host to trigger development of a nodule structure on a legume plant. And so they've grown up a whole lot of different fungi and shown these LCOs are present in a wide range of fungi. And using the pure chemical, they showed that these lipo-oligosaccharides affect spore germination, branching of hyphae, pseudohyphal growth, and transcription. And so Jean-Michel contacted me and said, excuse me, had I considered the possibility that when we made the deletions in the chitin deacetylases, instead of just affecting cell wall, we potentially might be affecting production of an LCO and signaling in the symbiosis. And that's a very interesting thought, and certainly one I should have thought of, because I worked on Rhizobium many years ago. And in fact, we showed that CDAB has considerable homology with NOD-B. So some interesting future work to pursue is to test whether CDAB may in fact be an ortholog of NOD-B, and rather than being involved in cell wall chitin deacetylation, may be involved in deacetylation of an LCO. I've retired now, but we have sent some samples to Jean-Michel to analyze, and in fact, they just arrived this week. And so we'll know very soon whether the CDAB mutant affects the synthesis of these potential lipid oligosaccharide signaling molecules. There's always more exciting things to do. And again, too late for me to work on them, but some fascinating future questions to address for others. And this has been a wonderful symbiotic system to study. And just to finish, some conclusions. Epichloe festucae can transition from free living to endophytic growth, and you see remodeling of the cell wall from chitin to chitosan. When it transitions from endophytic to epiphytic growth, you see remodeling from chitosan back to chitin. The genome has 3 chitin deacetylases, and our genetic analysis shows that 2 of these are very important. And in that very last slide, I've raised the question as whether one of these may in fact be important for lipo-oligosaccharide signaling as well as cell wall biosynthesis. And thanks there to a lot of organizations and people. I won't go through them all, but I've had very good support and collaboration with people all around the world over many years. It's just been fantastic. Thank you.
Thank you, Barry. I clap on behalf of everybody. That's the part we're missing, the in-person warmth. That's been fantastic. What great images. I'm fascinated. Everybody can type their questions. Pueden preguntar por las preguntas y respuestas o por el chat, por favor. And let's see, while people start asking questions, I have a question, because we know that, okay, there is a differential expression of genes in if the fungus behaves as an epiphyte or inside the plant, right? But yes. So I'm assuming outside there is polar growth.
Yes, exactly, there is. Yes, you're right.
So there's a switch from polar growth to this intercalary growth.
Yes.
And I'm just trying to picture how all the machinery, that is working at the apex, when it enters the plant, which probably it's entering the plant through a also polarized mechanism, switches to this intercalary growth.
Yes, exactly.
And even if there is no chitin in the cells that you're looking at, and you only see chitosan, the chitin has to be priorly synthesized, right? So chitin synthesis still have to go to those lateral walls.
Exactly.
Right?
Yes, you're right.
So I know it's very complicated, but do you have any idea on how this is happening?
You're exactly right. You first got to make chitin before you can get chitosan. And so if we're not seeing chitin in the cell wall at all, there is the possibility that it's masked, just when you're using these biosensors. But the fact that the septa, which are intercellular, are staining up, we don't think this is the problem, but there may be some masking. But I think with the intercalary growth that there'll be movement of vesicles to those septa, bringing chitin, and as the cell wall gets stretched, there must be very rapid conversion of the chitin oligomers to chitosan. But quite honestly, I don't have an answer for you, but you're exactly right. There's a dynamic balance between the chitin synthesis and the specific chitin synthetases that are, I think you've even done some work in this area of movement, the chitosomes to the tip of the hyphae with tip growth. That whole chitin synthesis must get changed and remobilized to the septa of the intercalary regions to set up the cell wall as it gets stretched and rebuilt as those hyphae grow and converted to chitosan very quickly. But yeah, there's a lot of work still there to do to sort that out.
See, I don't see questions on the chat. There you go. There is a question. It says, is the pathogen manipulating the plant defense system or the cell plants around it?
Certainly, there's a whole array of effectors, and many of these are differentially expressed when you compare the transcriptome in culture versus in planter. And while we don't have any direct evidence, it will be no surprise to me that the role of some of those effectors are in the suppression of host defense. And we do see this when we look at the plant transcriptome, that there is a suppression of host defense. But again, more study is required to really sort that out. And so it's kind of tricky, because while we talk about this being a symbiont, it also can be a pathogen at a particular stage in the life cycle. And so it's a developmental thing as well. And there's a very, very fine balance between the two, being growing in a very restricted sort of mutualistic way versus prolific growth.
Okay, there is another question from Jimena Carrillo. She loves your micrographs, and she's asking, what are the red dots inside the plant cells in the double mutant? We don't have the number of the slide for that, but in the double mutant, you were showing a signal that it was higher in the double mutant than in the single mutant.
Yeah, so I mean, this image is probably typical. That's a propydium iodide staining. So the big dots will be the nuclei of the plant cells, and the big yellow dots there are the nuclei of the plant cells. And if you zoom in on that, you can see the small yellow dots coinciding to the nuclei of the fungus.
I think she refers probably on the right panel that there are red. Jimena, you can turn on your microphone.
Yeah, sorry.
Yeah,
that one.
okay.
Exactly what, yeah, you just move your, yeah, those, thank you so much. Yeah, no, the previous one, over there in your arrow.
Are these red dots?
Yes,
here?
yes, thank you.
Yes, so the yellow ones are the nuclei. So what are the red ones? Oh, they must be chloroplasts, of course. So there must be chloroplasts lighting up in the plant cell, because let me just look at the size of them. Yeah, we haven't confirmed that, but no, that is kind of interesting. I think it must be picking up the nuclei of the chloroplast, or just chloroplast. Not entirely sure, but that would be my explanation, that these are chloroplasts we're seeing.
Yeah, it's just like the number is, at least to me, is increased in that one as compared with the other set. So it might be chloroplast, because I suppose it's the red is a chlorophyte, right? It fluoresces in red. So that's right. It is just interesting how at least I can see many more of those in that.
Yes, no, that is interesting. It's always tricky with microscopy, quantifying things. I think we would need to have a look, and she certainly, Nazanin, had many other images. We always try when we're publishing to put 2 or 3 panels that are representative of what we see, and we always try to quantify what we can. It's a wee bit difficult when you disrupt the actual growth, but there may be a difference here that we've overlooked, and it might be worthwhile just going back and seeing whether chloroplasts are affected in some way, but it may be that just coincidental that the images she's captured show more chloroplasts than in the wild type, for example. Yes, I mean, it's quite a big difference between the panel on the left of B and the right of B, isn't there?
Right.
Yeah.
Yeah.
Yeah. No. Good point.
Thank you. Thanks for your talk. Amazing. Thank you.
Okay, we have another question from Rufina Hernandez. She's asking if you know of any other similar systems for fungal plant interaction.
In terms of a symbiosis,
I guess, Rufina.
Yeah, because
there you go.
This is something very specific, and I mean, probably there is co-evolution of these the fungi with the plant. So I wonder if there is another system similar in, I don't know, in soils that doesn't have much water, or I don't know if there is this co-evolution is very common in plants we require the symbiosis or something. I don't know.
yeah. So this is very specific. So this group of fungi only infect the C3 temperate grasses. So you don't find them, for example, in the C4 tropical grasses. And there is some specificity between particular species and particular plant hosts. So they've evolved together over a long period of time, and their center of origin would be in the Middle East, where grasses evolved. There are some of these endophytes in some of the rye and sort of the wheat-related grasses, and there's a lot of interest in those. But in terms of other plant species, these certainly don't infect other plant species. but there are other endophytes, and one nice example I can give you is the cocoa plant. There's some work done by a group many years ago actually showing there's an endophyte present in the cocoa plant, and this helps protect the cocoa plant from certain plant pathogens. And I imagine there's lots of endophytes out there that remain to be discovered, and I think it's potentially a very fruitful area of work. I get a lot of papers on endophytes to review, but unfortunately many people don't do the microscopy to really show that the organism is growing within the plant. They do sort of sterilization tests and grow in a petri dish, and it's not a very good assay for demonstrating whether something is endophytic. You've really got to stain the leaf tissue and show these fungi are present within the plant, and then try and isolate and grow them. But I think there's huge potential for looking at endophytic fungi and all sorts of other plants.
We have another great question from Marco Buenrostro. He says about the stains that you're showing, that they are very general. How can you differentiate from other fungi that the plant might have, if there are any other endophytes, let's say, or is that not happening?
So these are from, we use seeds that have been grown in the field that are free of endophytes, so are free of Epichloe endophytes anyway. And before we use them, we surface sterilize. And so we always, and I haven't got any images here, we have plants that are uninfected. So remember, all these images that are shown are from plants that have been artificially inoculated with these strains. And so if you look at an uninfected plant, you might occasionally see some fungi on the surface, but you'll never see endophytic fungi in these plants. So that's the control, really.
Just a curiosity, is this confocal microscopy or is this epifluorescence?
These are stacks. These are confocal stacks. Right. Yes, okay. Yes.
We have another. Oh, go ahead. Go ahead.
I was just going to say I have to credit the students and postdocs in my lab. They're the ones that spend hours and hours sort of getting these nice images.
Yeah. We have a question from Gilberto Urlango. Go ahead, turn on the microphone, Gilberto.
Hi, thank you for your talk. Very nice images. And I'm just curious, when you mentioned wheat glutenins, I was wondering if lectins can be involved in that too.
Whether lectins can be involved in what?
As wheat agglutinins. When you mention wheat agglutinins, I'm just wondering if, do you think lectins can be involved?
I'm not, yeah, we use them really just as a probe for the chitin and the Elixiflor 488s attached to that. And as I understand, the wheat germ agglutinin is used because it's a rather small protein, and that allows for good infiltration into leaf tissue. So I think that's the reason that's used. But whether there are lectins themselves involved in the interaction, I really don't know, sorry.
That's all right, thank you.
Okay, so I think that was all the questions we had, yeah? Am I missing somebody? Let me know if there are any hands up. I don't think so. Well, thank you again so much for this beautiful presentation. Oh, is there somebody else?
Maricel, you missed me.
I'm sorry, I don't see you.
Oh, well, I did all I could here.
Go ahead, Dr. Varnicki.
Oh, I put tape on my computer.
Oh, there you go.
Hi, Barry.
Hello. Hi.
Good to see you. Is it good afternoon or good evening or what is it?
it's the middle of the day and it's a nice sunny day, and I've been up early outside of my garden. So, and it's lovely to see you and see you're still involved in science. You're a bit of a legend.
Yeah. Well. Yeah. But anyway, I have a question. And when you speak of intercalary growth, and you did it repeatedly, what do you mean by that exactly?
What I mean by that is the hyphae are laying down septa in the middle of the hyphae and not near the tip. So these hyphae are attached to the plant cell wall. They've been stretched, and then they're undergoing septation in the intercalary zone and not at the tip. It's exactly how plants grow, plant leaves grow in grasses.
Yeah, in plants it's true, but in fungi, I don't believe anybody has recorded intercalary growth. All growth is apical.
I think there's some examples in some of the basidiomyces when the stipe of the mushroom grows. I think some of that growth is intercalary, if I recall.
Yeah, coprinus. Yeah.
Yeah, it's certainly quite unique form of growth, and yeah, I mean all those cell polarization components like RAC and CDC 42 and 24 and all that machinery, it has to get mobilized to those intercellular sites to bring about this new cell wall synthesis. And it's very challenging to study because, of course, you can only study it inside the plant. Tina Voicey, I don't know whether you remember her, she's talked to this. She was trying to grow these hyphae on cellophane strips and stretching them mechanically to see if she could replicate the intercalary growth in the lab, with some success, but very difficult. So we need some better tools and some better markers to really dissect what's going on.
Yeah, you have to do autoradiography really to confirm this.
Yes.
One more point here. In our experience, chitin deacetylation is never complete. You know, and in fact, chitin is always has a percentage of chitosan. So it is possible that when you say there is no chitin, there is still chitin there. But the stains are really not completely reliable for this.
Yes, I totally agree with you. It's while I said that, I'm certainly well aware that the chitin gets processed from longer molecules to shorter ones, and I think you're exactly right. There'll be fragments of chitin still present.
Yeah.
Yes, totally agree.
Well, I enjoyed your talk, and I'm still alive. Okay.
Well, I'm still alive too, although my lab shut down now, which I'm not suffering too much withdrawal symptoms, but I must say I do miss it on occasions. It's been kind of fun doing this science.
Let's see, we have one more question. I think we'll let this one as the last one. Thanks.
Thanks.
There is somebody asking, how could we take advantage of the lipoketo oligosaccharides or chitin deacetylases to use their mechanisms in the control of plant pathogens?
Oh, wow.
That's a tough one.
Yeah, that is a tough one. I guess if these are indeed signaling molecules and they're required for the growth of, say, of a fungal plant pathogen inside the plant, then there may be inhibitors of these small signal molecules. I don't know that you could chemically inhibit them and then perhaps inhibit the growth of the fungus. what I will predict, and it's kind of obvious, is I think this is going to be a very active area of research, and not surprising, Nancy Keller's name pops up in association with John McCall at Wisconsin. So I think they're going to have some interesting data to present in the near future.
Okay, there is a last one, but I think we're out of time. Somebody's asking about inducing priming. Not sure what that means.
Oh, plant priming. Yeah, just yes, very quickly. Yeah, so we've had a collaborate. This Alexandra von Humboldt award was terrific. So I've had a number of collaborations with German scientists, and one was with Gunther Dollerman in Cologne, and we've just published a paper in what's it in? Anyway, there's a paper out jointly with Gunther on proteases, and not specifically on priming, but it does mention some of these early processes. The plant is responding to the presence of the endophyte. Experimental botany that's published in. So have a look at that.
All right. Well, yes. Now, yeah, we have to move on. We have another session, the last one of the day. So thank you so much again.
Yeah, well, thank you for the invitation, and lovely seeing you all. And just makes me a little nostalgic talking about the science again. First talk I've given in about 2 years.
Oh, wow. Well, you still give talks very well as usual. So have a good day and see you soon, Barry.
All right. Thank you.
Bye-bye.
Thank you, Gilberto.